All right, today we're getting into basically the electron transport chain and photosynthesis. So make sure you go over the objectives and all your key. So we're going to begin just like we did in the previous lecture with a brief discussion on energy. Now, in the parts of metabolism that we've learned already, so glycolysis and the TCA cycle, we've been collecting electrons and we've been putting them on energy carriers. And what we're going to do is transfer that energy by moving the electrons through an electron transport system or an ETS. You've also heard it called the electron transport chain or ETC. And the whole point of this is we're passing those electrons from protein to protein, and that movement of the electrons is literally generating electricity, and that electrical energy is enough to pump protons to one side of the membrane. So we're going to have a concentration gradient with a lot of protons on one side, and in the cytoplasm there's going to be fewer. And then we will allow those protons to pass through ATP synthase, or we'll use those protons for other things, and that will allow us to generate ATP, or we can use that proton motive force to power other parts of the work that we do in the cell. The whole process of the electron transport chain is a series of steps where we take an electron donor. This is usually a molecule that is in a very reduced form, and it wants to readily give up its electrons. And then we will pass that electron onto a molecule or a protein that is what we call an oxidized electron acceptor. Basically, it wants that electron a little bit more than the donor did. And then we keep passing this electron from molecule to molecule in order of increasing desire for the electrons. So we pass it from one that wants the electron but doesn't want it as much as the next one until we get to the final electron acceptor, which is oxygen. And oxygen is an incredibly strong electron acceptor. It rarely ever donates its electrons. So this is kind of a terminal point for it. And by breaking it down into these various proteins and creating the electron transport chain, we're pulling more energy out of that process, like we talked about before. If we did this all in one go, too much of the energy would be lost as heat. And so by breaking it down into smaller piecewise reactions, we can pull more useful energy out of these electrons. And we use that, as I said, to make our proton gradient to give us proton motive force. That's an energy source for other things in the cell. So there are three major classes in how prokaryotes acquire energy using an electron transport chain that we're going to go over. There's organotrophy or chemoorganotrophy. This is us. This is the one we're most familiar with. We take an organic molecule and we use that as an electron donor. So we're taking the electrons and the energy and the carbon from this organic molecule that we eat. But our terminal electron acceptor can be organic or inorganic. Oxygen is an inorganic molecule. So usually, if we're talking about humans, we are using that inorganic terminal electron acceptor. And then there's lithotrophy or chemolithotrophy. This is going to involve inorganic electron donors and can be organic or inorganic terminal electron acceptors, just like with the organotrophy. And then we have phototrophy. So this is where basically we're talking about light powering all of this. We're going to capture light, usually with some type of pigment like chlorophyll, and then use that to split either hydrogen sulfide gas or water, or we can use it to split organic molecules, and we take the electrons from those molecules that we're using light to break apart. So when we talk about these electron donors and electron acceptors, they are in oxidized or reduced states, and they're called a redox couple. So they're A complete redox reaction has two redox couples, one that accepts the electron and we say becomes reduced, and another that donates electrons or becomes oxidized. And there's another term here, the standard reduction potential. And what this basically is how likely is this molecule going to accept an electron or take an electron? So oxygen has a very, very high standard reduction potential, a very positive standard reduction potential. This means it's very readily going to take those electrons. Whereas when we look at NADH, it has a very negative standard reduction potential, meaning it wants to give up those electrons rather than take them. So the whole point of this is we're proceeding from something that has a very negative standard reduction potential to a very positive standard reduction potential. And we discussed this a bit yesterday, and I brought it up again now, but remember that all reactions require activation energy. They're also going to lose energy as heat, and that we break our reactions down into smaller pieces so that we avoid losing too much energy as heat. We want to keep as much of the energy that we're working hard to get to ourselves. Now, electron transport systems are built within a membrane. So you have to have a membrane in order for these to occur. And they're usually going to maintain an ion gradient where one of the aqueous compartments on the other side of the membrane has lots of protons, and the other side, the other aqueous compartment, does not. That way we can keep that proton potential flowing into the cell, and we can use that to power ATP synthase. Gram-negative bacteria, their electron transport system is in their inner membrane, the one that's close to the cytoplasm. And we can see that too when we look at mitochondria. It's not their outer membrane that this happens in, it's in the inner membrane. It's within those folds where all of this is going to occur. Now, eukaryotes do not have this in their external membranes. So a eukaryotic plasma membrane does not have an electron transport system. We rely on the electron transport systems of our mitochondria. So the membrane proteins that are embedded in the electron transport chain are called oxidoreductases. There's some other terms for them, but this is the one your book uses, so it's the one that we're going to stick with. These enzymes oxidize one substrate, so they remove electrons from it, and reduce another. They donate electrons to it. So we're coupling the different halves of the reactions. We're saying, you're the electron donor, let me take your electron and I'm going to pass it to the acceptor, and it's going to pass it to a new acceptor, and they are the enzymes that make this transfer happen. And so usually these enzymes are made up of multiple protein components, and they form a complex, and many of these protein components are what we call cytochromes. And when we look at that term, cyto means cell, chrome means color. So a cytochrome is just a colored protein in the cell, and we know of one that we used in lab, the TMPD that we used for the oxidase reaction. We were testing to see if the cells had cytochrome C oxidase. That is an oxidoreductase enzyme. And if it did, it would turn the cell blue in its altered redox state. So when it came into contact with this oxidizing agent, it caused the cytochrome to shift its color because it was absorbing a different part of the electromagnetic spectrum. And so these are just proteins that contain pigments, and pigments are just chemicals that can absorb light energy, and they do it at different wavelengths. We're going to get into a little bit of this near the end, but different wavelengths of light hold different levels of energy. So when we talk about infrared light, that's pretty low energy light, or red light is very low energy light. But blue light is very high energy. UV, gamma, X-rays, those are all very high energy wavelengths of light. So if we want to get a lot of energy from light, we need a pigment that can absorb those higher wavelengths, those things like blue or purple light. If we don't have proteins that can absorb those, we will be taking in lower energy light. So the way that this works is we're going to take the electrons from NADH or another electron donor and pass them piece by piece through our cytochromes to our final electron acceptor, moving from a low reduction potential to a higher reduction potential. NADH is one of the lowest reduction potential molecules, so it very, very readily wants to give up its electron. It very readily wants to be oxidized. And so then all that energy that's being released through the movement and transfer of these electrons is just there to power proton motive force. So we're making that proton gradient. Now, with this one, once again, the terminal electron acceptor has a very high affinity for electrons and does not want to give them up. Now, proton motive force, just a quick brief summary on it. I feel like we've talked about it a lot in this class, but let's actually have a slide dedicated to it now. So this is the transfer of hydrogen ions from the cytoplasm into the periplasm or into the external environment, and that's going to create an electrochemical gradient, meaning not only do I have a chemical gradient where I have hydrogen on one side but not on the other side of my membrane, but it's also an electrical gradient, excuse me, where I have many, many positive charges with those protons on the outside of the cell and not as many positive charges inside the cell. And so this creates proton motive force because now think of it as the dam. We've dammed up all of these protons and they want to flow down their gradient because we're fighting entropy right now. Nature says we want to be equal on both sides of that membrane, and they can't do that if they're being held to one side of the membrane. So as soon as a channel opens up, usually something like ATP synthase, they're going to want to flow down that channel and kind of even out the gradient as much as possible. And that flow, kind of think of water flowing down the dam, we capture that to turn turbines. That is literally what's happening with ATP synthase. We're letting the flow of the hydrogen ions turn a turbine in this enzyme, and that's going to generate energy for us. So that is the whole process, and we call it chemiosmotic theory. And it's the whole point of the electron transport chain. We don't really care about passing around this electron unless it's doing something for us important. And it is using that energy from those electrons, from that electricity that we're generating, moving them through the membrane to create this proton gradient. Now, proton motive force is not just there to create ATP, although that's a very big part of it. In bacteria, it powers flagellar rotation. In eukaryotes, we move our flagella differently. Bacteria, theirs is more like a propeller on a boat, whereas with eukaryotes, it's more like a side-to-side motion, like a tail on a fish. And we use ATP to power it instead of proton motive force. But every single cell uses proton motive force for many different areas that we do work in the cell. Things like taking up nutrients, so those antiporters and those symporters, we use proton motive force to power them. Bacteria will efflux toxic drugs out of things like the ACRAB-Tolc transporter, and they'll export them out so the cell doesn't get poisoned. Lots of interesting things. Now, while every single cell always uses proton motive force, there are some that we talked about way at the beginning, where if they are in a very alkaline environment, they will also evolve to use sodium transport, so sodium motive transport. Sodium ions are positively charged, so it kind of fulfills a similar role. However, those cells that use sodium motive force also still use proton motive force. It's just a very important thing to all cellular life. So let's get into this now. The electron transport system is going to use various carrier molecules and proteins, as I've already kind of alluded to. We're using mainly NADH and FADH2. NADH more readily gives up its electrons and can give up more energy in its electrons than FADH2. And they're going to transfer them to oxygen, which is our terminal electron acceptor, and make water as a result. This is going to be aerobic bacteria and mitochondria. So there are bacteria and organisms that do this slightly differently, but we're going to look at it right now in terms that we can understand, in terms we're more familiar with. There are three main components to a functional ETS, or electron transport chain. The first one is that oxidoreductase enzyme. It's also called a dehydrogenase. This is the enzyme that's going to first take the electron. So the donor, something like NADH, is going to give up its electron to this oxidoreductase. Then the oxidoreductase is actually going to start passing its electrons through what's called a mobile electron carrier. We also call this the quinone pool. They're a bunch of molecules. They are not proteins. They are lipid-soluble molecules that live inside the membrane itself, inside that fatty acid tail part of the phospholipid bilayer. And they can move around, and they can go anywhere they need to within the membrane. And they're going to accept the electrons and keep passing them around until we get to the terminal oxidase. And this is the enzyme that's going to give up its electron to the terminal electron acceptor. So it's receiving the electron from the quinone pool, which received its electron from oxidoreductase, which received its electron from NADH, which received its electrons from either the TCA cycle or glycolysis. And finally, it gives up that electron to something like oxygen, which is what we're going to focus on right now. So let's get into how this works, and we're going to do it step by step. So step one, the oxidoreductase is going to receive a pair of electrons, because remember, NADH carries two donatable electrons. And so it's going to take them from the organic substrate. NADH is an organic substrate. Or it can take these electrons from an inorganic substrate like hydrogen gas. And then that oxidoreductase is going to pass the electrons to the mobile electron carriers like a quinone. So these are the lipid-soluble molecules that are making up the quinone pool. And these ones are going to be the major factor that generates proton motive force. So the quinone is going to pick up two hydrogens from the cytoplasm and use that to reduce the quinone to what we call a quinol. And we're going to get into a little bit more of what this all looks like on the next slide, because we have a good picture there. But there's many different quinones. They all have their own different side chains, but we tend to refer them as Q for quinone and QH2 for quinol. So let's get into this here. Step 3 is where we oxidize NADH and reduce Q. It's a coupled reaction. So we take that NADH and we pass those electrons and start to reduce our quinones to quinol. And that whole process, we end up releasing 4 hydrogen ions from the cytoplasm into the periplasm or the external environment. So first, flavin mononucleotide, FMN, is reduced. It's going to accept the 2 electrons from NADH. It's then going to transfer those 2 electrons to these iron-sulfur clusters. So iron is usually in a charged cationic state, so it readily wants to accept electrons. And so these clusters will take on the electrons and cause conformational changes in these many different alpha helices in the protein. And these alpha helices will have a shape change that is just enough to take a hydrogen and allow it to flow out and basically forces it out. So they're going to drive that movement of the 4 protons across the membrane. And this is all because they're taking these electrons and passing them one to another. The iron sulfur clusters are embedded in this area of the protein, the oxidoreductase protein. Then they are going to give up the 2 electrons to the quinone pool. And the quinone pool is going to do that and take up 2 hydrogen ions at the same time to become quinol. Because we can't just take electrons, we want to usually balance that out. And remember, the hydrogen ions are protons. So if we take 2 electrons, I want to balance it out by taking 2 protons as well. And that gives it that QH2 structure that we discussed. Now, part 4, that quinol, the QH2, it's going to give up its 2 electrons, and it's going to give it up to this terminal oxidase complex, which is a protein. And this is the part that contains the cytochrome. So this is the part that has the color changing chemicals. And when the quinol gives up its electrons, it's going to convert back to a quinone. And when that happens, the 2 hydrogens that it picked up from the cytoplasm get sent out into the periplasm or external environment. So it's feeding more of those hydrogen ions to the side where we're going to keep them dammed up. So we're creating more and more of a concentration gradient. And then what we end up doing is continuing to pass those electrons from cytochrome to cytochrome, basically. And while we do that, it generates enough energy to move another 2 hydrogen ions across the membrane. So we're just pumping out these hydrogen ions as much as possible here. So the terminal oxidase complex has now passed that electron from cytochrome to cytochrome within this structure, taking out as much energy from that electron as possible and using it to pump those 2 more hydrogen ions to the other side of the membrane. And finally, we're left with an exhausted 2 electrons, meaning they're now in a very, very low energy state. These electrons cannot go to just anyone. They have to go to a molecule that is truly desperate for oxygen, one that is a very, very strong electron acceptor, like oxygen. And so the oxygen will pair up the 2 electrons with 2 protons, and it'll form water, which is released into the periplasm or the environment. Now, in E. coli, the electron transport chain pumps out 8 protons for every NADH molecule created, but only 6 for every FADH2 created. And this is just because FADH2 is not quite as strong of an electron donor as NADH, so it takes lower energy electrons and has less energy to give up, basically. So we can't use that work to move as many protons over. So this is basically the whole thing in one picture. From the TCA cycle or glycolysis, we get NADH. That NADH is going to give up 2 electrons to this first oxidoreductase. That's going to get passed to the iron sulfur clusters, which are going to cause conformational changes in this protein. When that happens, it forces these 4 hydrogen ions across the protein and into the periplasm or environment. And then those 2 electrons get transported and placed onto a quinone. This causes the quinone to take up 2 protons, 2 hydrogen ions to become a quinol. We then pass the electrons throughout this quinone pool. And finally, when the quinone pool is done and is ready to give up those electrons to our cytochromes, and our terminal oxidase protein here, it's going to release the 2 electrons and take those 2 hydrogens that it picked up at the beginning and pump them out. Then the passage of the electrons through this protein complex is going to pump out two more hydrogen ions. And finally, when the electrons are completely exhausted, they combine with water and oxygen and hydrogen ions to make water. And so we have 2, 4, 8 that can get hydrogen ions that get placed into the periplasm or the environment from 1 NADH. There's a whole video of this. If you're someone that needs to see it actually like moving and working through it, I highly recommend you watch it. It should already be posted. So let's talk about what we end up doing with all of that proton potential. One of the things we can do is drive ATP synthesis. And we do that using the F1, F0 ATP synthase complex. And this is a very fascinating enzyme, very complex enzyme, and it's made of two parts. The F0 is the one that is embedded inside the membrane itself. And this is actually the part that's going to spin because of the protons flowing down that gradient, just like a dam. So with a dam, we move the water from one side where it's all walled up and let it flow down. And through that flowing process, that energy is used to spin a turbine, and that turbine is what's going to then create and generate electricity and power the whole town. So, kind of the same thing here. The hydrogen ions flow down, it spins this turbine, and then we have an adapter, this gamma subunit, that's connected to a bunch of alpha and beta subunits. And this whole portion in the cytoplasm is called the F1 part of the complex. And this rotation of the turbine is going to power conformational changes between the alpha and beta subunits that basically catalyzes ATP synthesis. So the power is from the hydrogens flowing and spinning this turbine. That turbine then sends its energy down into the F1 complex, where it causes conformational changes in the subunits to basically take an ATP and an inorganic phosphate and pop them together, and that makes ATP. So we can see that all happening here. The protons flow in through this part of the subunit, and this whole part spins, indicated by this arrow. And then when we look down here, we have an alpha and beta, and then there's like these channels in between an alpha and beta where a ADP and a proton can come in, where the reaction gets catalyzed, and where the ATP can then leave the cell. So entering in is the substrates, and then we catalyze it, it forms ATP, then ATP is able to leave when that conformational change happens again. It's very fascinating overall. I'm not going to keep repeating it here because it's just basically everything that I just said, but I highly recommend you watch the video because the whole process is quite fascinating to watch, and it's a short little video. Overall, this can make 34 ATP per glucose. Now, this is a theoretical maximum because not all of the energy in that glucose gets converted into NADH to power proton motive force and to create this ATP from ATP synthase. We can use the energy from a glucose to do other things. We can make something like NADPH through one of our alternative glycolysis pathways. And when we do that, we're going to use that for biosynthesis rather than ATP generation. So theoretically, this whole process from one glucose can give 34 ATP plus the 2 ATP from TCA cycle plus the 2 ATP from glycolysis. That's how we get that 38 number of ATP yielded in central metabolism from one glucose. So here's the video. Just a reminder, it's posted on Canvas. Highly recommend you watch these ones. They're actually pretty good. All right. So that was all aerobic respiration, where oxygen is present to act as the terminal electron acceptor. But life didn't evolve to live in oxygen, as we've discussed before. In fact, the first life evolved in a very oxygen poor environment. There was hardly any molecular oxygen that they were being exposed to. So the first type of respiration to occur was actually anaerobic respiration without oxygen. Now, obligate aerobes are ones that have to use oxygen as the terminal electron acceptor. This is us. This is most plants, or this is plants, excuse me, and some bacteria. But prokaryotes, the archaea and the bacteria, they can do some fascinating things, and they have a wide range of terminal electron acceptors that they can use. And as a result, this is anaerobic respiration. Usually it's going to happen in areas where there isn't a lot of oxygen available, because if there is oxygen available, it's the best electron acceptor. You would want to be able to use that, because otherwise it's all this energy that you're not going to be able to tap into. So if you live in an area like a wetland, or deep in the soil, or in the human digestive tract, or in deep sea vents, you're not going to be able to use oxygen because there's not a lot of it present. And so you have to do this anaerobic respiration. So one terminal electron acceptor that can be used is nitrogen. And what happens is, as we take the electrons, we'll take them from a form like nitrate or nitrite, and we will slowly oxidize it until we get nitrogen gas. So as a result, this is taking bioavailable forms of nitrogen, ones that other organisms can use and assimilate, and it's converting them into forms that are not available for life to use. So we call it dissimilatory denitrification, because we are denitrifying the soil. We're taking nitrogen out of the soil and putting it back into the atmosphere, where it's not really accessible to life, unless you're the bacteria that can do the opposite conversion. Then we can also use sulfur. Now, sulfur, they aren't as good to use as nitrogen, but if you don't have nitrogen or oxygen available, or you can't use them, sulfur is another really good choice. And so this we see a lot in the ocean. Arctic waters, thermal vents, basically everywhere in the ocean, we tend to see a lot of anaerobic respiration using sulfur as one of those terminal electron acceptors. And as it is reduced, we can see this whole pattern here, where it goes from sulfate to sulfite, all the way to either elemental sulfur or the very disgusting smelling hydrogen sulfide gas. Um, and very interesting is that sulfate is the most prevalent ion in the ocean. It's very low energy, but it has high availability. So in the end, even though you're not getting a lot of energy from it, the cell says, yeah, but there's so much of it around, I'm never going to starve, so I might as well use that. Then we got to talk about lithotrophy and something called methanogenesis. So with lithotrophy, this is where we get energy by oxidizing inorganic electron donors, something like hydrogen or iron. Um, there are some organisms that are obligate lithotrophs, where they can only oxidize molecules like this. However, most of the oxidoreductases can do both. So most of them can oxidize these if necessary, and they have evolved to be able to oxidize organic molecules as well. So we kind of see it as these lithotrophs were kind of the early form, and then we evolved a new ability on top of that. So nearly all lithotrophs are bacteria or archaea. You don't see this a lot in the eukaryotic domain. So there's another form of anaerobic respiration, where they can actually use iron oxidation. Um, this bacteria here, acidithiobacillus ferrooxidans, sometimes these words are crazy. This bacteria takes iron in the Fe2 plus form, that ferrous iron, and reduces it and does an alternative electron transport chain as a result. Now, iron is a very poor electron donor, but they still can use it if necessary. And in the end, a lot of times this doesn't occur at pH 7. It has to be at a very low pH environment in order for this iron oxidation to be a viable method for the cell. Methanogenesis is where they reduce CO2 to methane. So we're taking CO2 and we're adding hydrogens to it until we form CH4, which is methane. So this is performed only by archaea, and they're called methanogens. And what they do is they'll often provide a unique niche for other organisms. So methanogens use methanogenesis to produce a bunch of methane, and then you have organisms that are called methanotrophs. Troph kind of refers to your eating, that can actually take this waste product, the methane, and eat it and use it to make energy. So just kind of an interesting um, little creation of a niche there. And then there's phototrophy. So this is all the different ways that organisms can harness the energy in light to power their cell. Usually what they do is they use light energy to power up the electrons. They give electrons an energy boost so they can capture more energy from passing that electron around. Now, a lot of us think, oh, we, oxygenate the planet using things like plants and trees, and that's what drives most of the oxygen production on Earth. While that is very not true, um, while they're obviously very important, much more than 50% of the oxygen formation on this planet is due to photosynthetic organisms in the ocean, most of them microbes. So without the microbes in the ocean, we would not have nearly as much oxygen as we would like. So that's why a lot of times you hear conservation efforts discussing terrestrial biomes, which are obviously incredibly important for other reasons, but we don't really discuss the maintenance of aquatic biomes, especially the ocean, as much when they have just as much, if not a greater, impact on some human health. So let's talk a little bit about how this all happens. So we're jumping now into a little bit of photosynthesis. So what we've been talking about with metabolism is a way to take food, take as much energy from it as possible, and convert it into proton motive force and an ATP. Now, when we talk about photosynthesis, yes, we are going to generate some ATP from this, but a lot of times when we look at photosynthesis as a whole, the purpose of it is to take this energy and create ATP and biomolecules that we can then use to eat later on. So we are looking at this more as a way to build up a pool of molecules that we need to do metabolism later. So with this, as I said, we are using light to excite an electron, and we are going to then be able to steal more energy from that electron. And usually we're absorbing the light energy through a pigment called chlorophyll. And this one reflects a lot of green light. It absorbs light in the rest of the areas of the visible spectrum. So that's why we tend to see these things as green. Now, this photoexcitation leads to something called photolysis. So this is light lysis, or breaking due to light. And what we're doing is using light to take something like water or hydrogen sulfide gas, and we are breaking these molecules to get at their electrons. We're forcing them to give up an electron to us. And as a result, it'll create oxygen or sulfur gas, and it will release protons as well. So with this, the electron that is released from the water or the hydrogen sulfide is then transferred onto an electron transport chain. And through this, the cell will create NADPH instead of NADH. It also creates a proton potential. So we are making proton potential for proton motive force. We can make ATP, and we're making the stuff that we need to do biosynthesis or to create new molecules for our cell later on. So this is not the completion of the breakdown of a molecule. This is the energy gathering stage to prepare us to make more molecules. So just keep that in mind. Basically, we can kind of see this whole process of photosynthesis as a reverse of everything we just learned in metabolism. We're doing an electron transport system first, then the Calvin cycle, which is kind of like a reverse TCA cycle, and then we're using that to generate something like glucose that we can use for energy. We can put that through central metabolism and burn it for energy, or we can use it to build up the structure of the organism. Now, let's dive into this a little bit more. There's a lot of different kinds of photosynthesis out there, but all of them share a few things in common. They need an antenna system. They need a way to capture the light. And so this is where they'll have a reaction center complex that has a pigment that can capture that light and excite the electron for the cell. Then that electron has to go through an electron transport system and generate energy carrier molecules. So you have probably heard of photosystems 1 and 2 in the past. And basically what we're going to discuss now are the different subgenres, you can say, of photosynthesis. Bacteria can do photosynthesis using just photosystem 1 or just photosystem 2, or the oxygenic Z pathway, which contains both photosystems 2 and 1. And this is the one that you are probably the most familiar with through the photosynthesis you've learned in previous classes. Now, photosystems 1 and 2 on their own are anaerobic. They do not generate oxygen. Only the Z pathway does. And the parts of photosystems 1 and 2 do share a common ancestry with each other. So let's get into photosystem 1. This alone, on its own, not paired with photosystem 2 for the oxygenic Z pathway, we're saying these are organisms that just have photosystem 1. And they're going to be the chlorobia, or green sulfur bacteria that has this. They use hydrogen gas or hydrogen sulfide gas or some other organic molecules, so they can use inorganic or organic molecules as their electron donor. Some have even been found to use iron as an electron donor. Not a very good one, but it still can donate an electron if necessary. Now, this photosystem is absorbing light at about 840 nanometers. This is not visible light anymore. This is effectively heat. They are taking infrared radiation and using it to power this photosystem. But as a result, infrared does not contain as much energy as the visible spectrum. So we're not able to get quite as much energy out of this light. We're not able to excite that electron quite as much as if we had used a lower nanometer wavelength of light. So the electrons are taken from these donors and ultimately transferred to either NAD or NADP. And this is going to be the energy that we use for carbon dioxide fixation and biosynthesis, so making molecules in the cell. Now, sometimes, not always, but sometimes they will use photosystem 1 to drive a proton gradient and drive ATP synthesis, but for the most part, this is more concerned about generating NADH and NADPH, mainly NADPH. Now, photosystem 2 alone is found in our alpha proteobacterial or purple non-sulfur bacteria. What these are going to do is they don't have an electron donor. So they take electrons from the bacterial chlorophyll itself. So they give up an electron from the chlorophyll, but then they have to return that electron back to the chlorophyll, or you'll have an unstable chlorophyll molecule that can't be used again. So they don't have an outside electron donor. This is a cyclical nature. The electron gets excited and then falls back to the same molecule. They transfer that electron to an electron transport chain, though, and we're able to get some energy from that to produce ATP. So this is called cyclic photophosphorylation. It's cyclic because we are releasing the electron and returning it to the same molecule. We are generating some hydrogen ion gradient, which will then power ATP synthesis, so phosphorylation of ADP. So it's cyclic photo from light phosphorylation. Now, they use light that's even lower energy, 870 nanometers. This is too low to generate NADPH. You need a little bit higher energy to do that. So between the cell needing to give back the electron and the fact that they're using such low level energy light, they have no direct way of making NADH or NADPH for biosynthesis. This is primarily used to make ATP. So we still will need electrons in a different way to create NADP for biosynthesis, and they will do this by eating. Really weird to think about. But if you consider it, we need carbon, we need energy, and we need electrons. So they are able to get their electrons from very, very low energy light. And unlike plants, that's not enough to sustain them and actually feed them as well. So they're saying, I'm not living in an area with high light, so I'll take whatever light I can get, but I'm going to supplement it by eating. And so these are our photoheterotrophs. They're going to use light energy for their electrons, and they're going to, or I'm sorry, they're going to use light energy to get their energy, but they're going to get their electrons by eating them from organic molecules, because there's no electron donor for photosystem 2. Just kind of crazy to think about. And then we have oxygenic photolysis. This is known as the Z pathway, basically because there are two instances where light excites the electron, and this kind of makes the electron move in what looks like a Z pathway through these proteins, these membrane proteins. So this is going to include homologs, not exactly photosystems 1 and 2, but things that have evolved from photosystems 1 and 2. And they're both going to be used together. So if we remember, photosystem 1 was our biosynthetic pathway. We are getting the electrons to make NADPH and prepare for carbon fixation. Photosystem 2 was, I'm going to eat my food to get that, but I'm going to use this light energy to power ATP synthesis. So when we put both together, we're going to get ATP, and we're going to get the stuff we need to do biosynthesis. So with this, 8 photons get absorbed, and 4 hydrogens and 4 electrons are ultimately removed from water. So we use water as the electron donor, and this is going to produce oxygen. That's why it's the oxygenic pathway, oxygenic photolysis. Then we're going to form 3 ATP, 2 NADPH, and 2 waters are going to be produced, or 2 oxygens are going to be produced. And then we are going to take the ATP and the NADPH that we make and fix CO2 into biomass. So as I said, the Z is because of the way the electrons are being excited. We start with photosystem 2, and it's going to absorb light in the 680 nanometer range, so it's already getting more energy. And it's able to strip 2 electrons from water with all of that energy that it's taking in. And then we make ATP because we pass it through the quinone pool, and we make that hydrogen ion gradient that feeds into the ATP synthase. Overall, it pumps 12 hydrogens across the membrane to power the ATP synthase. And then it's going to transfer those electrons to plastocyanin, which then transfers them into photosystem 1 here. Because remember that photosystem 1 requires an electron donor, so it's getting that from the original water that we took the electrons from. And then in photosystem 1, we are taking in 700 nanometer light. So now not quite as much energy, but still really high energy visible light to excite the electron again. And because of this, it's ultimately able to make NADPH. So we get ATP synthesis and NADPH synthesis. And there's a whole video about that if you are not familiar with photosynthesis. And that is it for today's lecture, guys. Please let me know if you have any questions. Otherwise, I will see you all next week. Remember that on Monday, I'm going to hold office hours at 9 a.m. So you can come on in early and ask me whatever questions you have. All right, see you guys later.